Hello! In this video I will introduce you to the ArtsDB web database and show you how to use it step by step. ArtsDB is a comprehensive database containing pre-calculated ARTS analysis results for bacterial genomes. This database is based on a target-directed genome mining approach and provides researchers with powerful clues for discovering new natural products by linking potential antibiotic targets with biosynthetic gene clusters. Currently, ArtsDB contains over 27,000 high-quality RefSeq bacterial genomes and over 43,000 genomes derived from metagenomes. The database offers two main search features. The first one is query building. You can perform organism or gene-based queries according to ARTS criteria by applying extensive filters. And the second is target-oriented search. You can query a specific target gene and analyze its distribution across different bacterial groups and its relationship with ARTS criteria. In this video, we will take a closer look at how to use ArtsDB effectively by demonstrating both search methods with examples. Now, let's move on to the first search option, query building. You can access this section by clicking on the Query tab in the top menu of the page. This option allows researchers to perform customized and detailed filtering on ARTS analysis results. In the first step, we need to select the genome pool to be queried. There are two options here. RefSeq represents a pool of high-quality bacterial genomes published by NCBI. Genomes from the Earth's microbiomes contain assembled genomes derived from metagenomic data. In this video, we will proceed using the RefSeq genome pool as an example. After selecting the genome pool, four main query categories appear. First, summary results is used to view the general ARTS statistics of genomes and is particularly useful for discovering interesting genomes for targeted analysis. Second, core hits allows you to identify core genes that meet one or more of the ARTS criteria: duplication, BGC proximity, horizontal gene transfer, and known resistance targets. The third category is dub hits. You can examine how frequently the selected gene is duplicated across different bacterial phyla. Lastly, BGC hits helps analyze the distribution and functions of important genes across different BGC types. For example, it is possible to filter NRPS clusters containing a specific gene. Each query category offers different search options within itself. After selecting the relevant category, you can access these filtering options by clicking the Add Term button on the right side. Each query type can be further customized with different combinations. In this section, we will perform a sample BGC hits query and discover how to search together. First, we will select the RefSeq genome pool. Then, we will add search criteria to our query by clicking on the Add Term button. In this section, the user is offered 9 different filtering options. You can also see these in the pop-up that opens under the search options. These include taxonomic searches such as phylum, genus, organism, and assembly ID. BGC type, you can specify the type of the cluster such as NRPS, PKS. Tiger fam model, description, and function. You can query based on the definition, model, or function of the core gene within the BGC. When adding each filter, you can use the AND or options to perform more narrow or broader searches. In this example search, we will try to find cases where the LSPA gene in genomes belonging to the Actinobacteria phylum appears as a core gene in NRPS type BGCs. We will enter the terms one by one. First, we enter Actinobacteria for phylum. We enter LSPA in the description section. Then, we simply enter NRPS for BGC type. Finally, we select the core option for hits type. When this query is run, only results that meet these criteria are returned. What does the results page show? Now we can quickly review all the results. Assembly ID directs you to the detailed ARTS page for the relevant genome. Cluster displays BGC details from the anti-smash analysis. Model name displays statistical and phylogenetic information about this core gene. It also provides a link to a target-oriented results page. BigFam GCF displays the gene family to which the BGC belongs via BigFam. It is also possible to access descriptions, functions, and taxonomic information. We can see 180 entries in this search result. To further refine, customize, and separate the relevant entries, we can use the search bar at the top of the results table. Let's narrow down our results by the Streptomyces genus. Typing Strepto will suffice for this example. We can then see that the results have been reduced to 5 entries. Additionally, the results can be interactively used with features such as sorting and exporting. You can download the results using the Export Selection button. After the query building search option, we can now move on to the second search option, Target-Oriented Search. You can access this section by clicking on the tab in the top menu of the page. The second basis search option in ArtsDB, Target-Oriented Search, focuses on a specific core or housekeeping gene and shows how this target is distributed across different bacterial groups and how it is evaluated according to ARTS criteria. When the page opens, a simple search field appears. You can perform a search by entering the model name of a target gene here. Alternatively, you can select from the system's predefined model list. Searching for the GAIR-B gene. In this section, we will use the DNA gyrase subunit B, GAIR-B, gene as an example. When we type gyrase into the search box, the system lists the relevant models. We select the DNA gyrase B subunit model from this list and then click the Insert button to search. We select DNA gyrase B subunit model from this list and then click the Insert button to start the search. Once the search is complete, general information about the selected gene model is displayed first. This information includes the ARTS reference set to which the model belongs. Further down, we encounter various graphs showing how the gene was evaluated according to the ARTS criteria. The first section of these graphs is the distribution of BGC proximity hits. The graph on the left shows the BGC types, for example NRPS or PKS, in which this gene is found. The graph on the right shows in which bacterial phyla this gene is found near a BGC. In this example, it can be seen that the GAIR-B gene is mostly found in NRPS-like clusters and in the Proteobacteria phylum. The third graph, in distribution of duplication hits, provides information about the replication rates of the selected gene across different phyla. This is particularly useful for understanding whether the gene has been repurposed for secondary metabolite production. In this example, it is observed that the GAIR-B gene is present with more copies in the Actinobacteria phylum. The final 2 charts on the results page evaluate the extent to which the GAIR-B gene satisfies the ARTS criteria. These graphs allow researchers to evaluate the potential of a given target to represent a resistance mechanism within a specific phylum. Once more, Actinobacteria demonstrates the highest number of significant associations in both plots. Finally, additional links related to the target gene are provided at the bottom of the page. The drug bank link shows drugs associated with this gene and their mechanism of action. The MyBig link provides direct access to known BGC examples where the gene is present. This information is highly valuable for evaluating whether a target gene has a potential resistance mechanism and for pre-selecting targets to be experimentally tested. In this video, we introduced the ArtsDB platform and explored 2 different search options. Query building, which allows you to perform detailed filtering with genome-based queries, and target-oriented search, which allows you to perform analysis based on a specific target gene. ArtsDB provides users with a powerful starting point for target-oriented genome mining studies. It is particularly useful for discovering new natural products, identifying potential antibiotic targets, and researching resistance mechanisms. In addition, its user-friendly interface makes it an easily accessible research for both researchers and students. Be sure to try ArtsDB to discover organisms, genes, and BGC structures suitable for your own project. Thank you for watching and good luck with your genome mining adventure.